So, welcome to the Software industry meeting. As you would guess from the title of the meeting, this is mainly for industry interests, but of course it's also open to other participants. And what we're going to do is give you a bit of an overview of the strategies, the approaches we used in Software, and as you will see, which will also be used for a follow-up project. So my name is Tom Gressner. I'm based at the University Hospital in Tübingen, where I lead the Rare Disease Center, but I'm also the coordinator of SOLVAD. So what is SOLVAD? SOLVAD is or was an EU-funded research project. It started in 2018 and will be ending end of June. Well, no, it has been ended, so that the date is not correct, has been ended end of March this year. So we had 22 partners from 10 countries, and as I said, it was coordinated here in Tübingen. We had actually one aim, was really to find disease causes for unsolved rare diseases or unsolved rare disease patients, and for those, of course, then the respective disease-solving variant. So when we talk about unsolved, for SOLVAD, it meant that we have patients with a respective suspected genetic rare disease without a molecular proven genetic diagnosis. And also the patient we included into the project had to have an exome sequencing and, of course, then an inconclusive exome sequencing. So we built a European or even a pan-European and well, going beyond Europe SOLVAD network, which consisted mainly of European reference networks, but also, of course, added respective bioinformatic patient organization and further expertise. So we started out with 4 ERNs, which are, so ERN is for European Reference Network, so ETACA, intellectual disabilities, rare neurological diseases, neuromuscular diseases, and tumor risk syndromes, and added a bit later 2 further on rare autoinflammatory and autoimmune diseases and rare and complex epilepsies. And we also collaborated in the project with 2 undiagnosed rare disease patient programs. And from the numbers underneath, you see that it was really a large network we have set up. So what are you going to do in this meeting? So I'm right in the welcome already. So after this welcome, this intro part, I will hand over to Sergi Beltran from CINAC in Barcelona to talk about the reanalysis approach, which is one of the main approaches we used in SOLVAD. Then the second part will be on the solvatin concept, and that's actually covering the second main approach, which is innovative omics technologies we used. So I will give a short intro into the solvatin concept we have developed in SOLVAD, and then we'll be giving you 2 examples, one from the RNA-seq and multi-omics perspective, and then covering the long-read and optogenomic mapping perspective. Then you can ask questions, and in the end, I will give you, as already indicated, a bit of perspective how we're going to continue and maintaining what we established in SOLVAD. And with that, I give the stage to Sergi.
Hello, good morning or good afternoon, since it's past 12 already. Thanks a lot, Tom, for the introduction. I'm Sergi Beltran. I lead the bioinformatics unit at the National Center for Genomic Analysis in Barcelona. And as Tom mentioned, I'll mainly be talking about next slide. Sorry, I'm going to be mainly talking about one of the approaches that we've developed in SOLVAD, which is the reanalysis of over 20,000 data sets. All of the results I'm going to show you is from the first 10,000 data sets. And afterwards, Vicente and Alex and Anna we'll talk about some of the analysis of novel omics data, new data that we have generated in the project. So I'm going to be focusing only on data we collected that already existed and that from which we have derived diagnosis through reanalysis. Next slide. In SolverD, we divided the participants, patients and relatives in 4 main cohorts. And these cohorts were at high level defined by the participating ERNs that Holm described a minute ago. And the reanalysis approach focused mostly on the unsolved cases, cases for which we already had at least an exome, but in some cases we actually already had a genome, and in which, even though this exome and genome and other tests had been done for these patients, the result was still inconclusive, and they did still not have a molecular diagnosis. That's cohort 1, and that's the one I'm going to focus on. But we also had other cohorts in the project. We had some cohorts that were specifically developed or proposed by the ERNs, for which we generated quite a few of data, mostly whole genome sequencing. Then the third cohort was what we call the ultra rare diseases, and these were phenotypically special or remarkable patients that, in this case, might not have an exome. And in this case, we did provide exome sequencing, and for example, in Genturis, we did these at deep levels, so we could account also for somatic mutations. And then the fourth cohort were the unsolvables, where these are highly recognizable clinical phenotypes or diseases that, despite having done before in the past some exome or genome sequencing, still we don't have a molecular cause for those. And for those, we decided we would apply all types of omics tools to try to identify some of those. But I'm not going to talk about those. I'm just going to talk about cohort number 1. Next slide, please. This is common to all the cohorts, though, and this is how the data comes into the SolverD infrastructure. Basically, we are recording all the phenotypic data to a system we developed in Barcelona, which is called the PhenoStore, that uses standardized vocabularies or ontologies such as HPO, the Human Phenotype Ontology, or ORDO, the Orphan Rare Disease Ontology, and we can intake this information either with Excel files or with JSON files, with phenopackets, or if you have not too much data, you can also enter this information manually through a user-friendly interface. And then also the genomic data comes in through the system, the RDConnect Genome Analysis platform. We process it with a standard analysis pipeline, and then we deliver this to the EGA, the European Genome Phenome Archive. That serves for us for mainly 2 purposes. The first one is a temporary space that we can use then to distribute the data to other partners, other clusters in the project, but also for the future, for archival and controlled access of this data for other parties that might want to use this collection of data from SolverD. Within SolverD, we analyze then the data through the RDConnect system, but also we analyze the data through many different methods that we run in either authorized local clusters or in a small cloud instance that we call the sandbox run in Groningen. And then also we have a system which is called RD3, Rare Disease Data About Data, which is mainly a database with an interface that we use to control all the different data sets, experiments, and analysis that we have done to the different data sets collected in SolverD. Next slide. So all this basically creates a resource that we believe it's going to be useful for the entire community, and it's going to be available at the EGA for download, but also available at the RDConnect GPUB for analysis through a user-friendly interface. When we have all the data released, that will include over 23,000 individuals and over 100 million unique variants. Next slide. The way that we have organized the analysis in SolverD is through a 2-tier or 2-expert approach. On one hand, on the left, we have what we call the Data Analysis Task Force. There is one such task force that brings together the bioinformaticians, the experts on data analysis. And on the right, we have several DITFs, Data Interpretation Task Forces. In SolverD, we had one the ITF per ERN. And this brings together the clinical experts that know about the diseases and about the genes that might be causing disease. And altogether, we worked on analysis plans through working groups to then systematically analyze the different cohorts and data sets in Solvar-D. The EITF generates results, generates variant files or lists of shortlisted variants, and then the DITF goes and interprets these variants and goes back to the patients or to the referring clinician and then confirms if one of these variants is indeed the one causing the disease in every specific patient. So a big aim here is to be able to return results to the patients. Next slide. Within this two-level expert scheme, we organize ourselves in working groups. These are more technical working groups, technology-based, and this applies, this is a very old slide, but this applies to both the data that we collected that was already existing and the data that we generated from scratch. And as you can see, we have, for example, for genomes, we had working groups on SMBs and indels, on CMBs, on relatedness analysis, de novo mutations, meta-analysis, but also working groups that were targeting other types of information, such as epigenomics, transcriptomics, somatic mutations, structural patterns, and so on. So it's kind of an ad hoc distribution of expertise, technical expertise, depending on the analysis that we have to perform on the data sets. Next slide. So these are the results that we have under review, and this is the first, I think, 2 data freezes. This accounts for about 10,000 data sets collected, exomes and genomes from already existing data sets that we have collected in Solvar-D, and this accounts for a total of 6,000 families. Once we also took out the data that had low quality and duplicates and so on and so forth. Here, important step is that we harmonize the data by using the ontology I mentioned before. We harmonized analysis. All the data went through the same set of pipelines. And then we went through this 2-level expert review in which the ATF and the ITFs, each of them applied their thresholds and went back to the clinics to see if these match the phenotype or not. On average, we think that for the very few variants that we return for every patient, it takes about an hour per case, more or less, to evaluate the results by the interpreters. The results from these first 6,000 families were that we were able to diagnose in total 12.5% of the cases. 8.4% of these cases were diagnosed through this systematic reanalysis of the data, in which we went through a very systematic approach in all the steps. But also we had about 4% of cases that were diagnosed through what we call an ad hoc expert review, which means that in parallel while we were doing the work in Solvar-D, the clinicians also reanalyzed their data and reinterpreted their data, and some of them then also identified variants on their own. It doesn't mean that these variants are also not in the systematic approach, but we wanted to keep these two outcomes clearly separated. Next slide. So as you can see, most of the results that we get, or most of the diagnoses, are still due to coding SMBs and indels that we had initially missed, or that they are from genes that before they were not known to be associated with a certain disease. But also we have many other types of variants that maybe are not routinely assessed, or they are more challenging to see in the data, such as CMBs. You can see that's the second category with 8.7% of cases diagnosed thanks to CMBs. But also we have quite a few SMBs and indels that are affecting splicing. We have also variants in the mitochondrial DNA, which is not always captured in exomes, but usually captured well in genomes. And in exomes, if you have probes to capture also, it's available there. But also other types of structural variants and mobile element insertions and short tandem repeats. So you can see it's a mix of different variants that went into this set of diagnoses. Next slide. Obviously, I don't have time to go here across many of the examples that we identified, but I'll just give you 3 here on interest of time. This is a case from Ithaca, 24-year-old male with facial asymmetry, autism, seizures, lower limb asymmetry, and described in the system as having underdevelopment of the left side And when we did the reanalysis of the data in SOVRD, we were able to detect a variant in PIK3CA in only 23 of 179 of the reads in that position. And this is a very low frequency at about 13%, and this is actually a rare de novo mosaic variant. And this also now is already in ClinVar identified as a pathogenic variant, and this allowed us to change also the clinical diagnosis and to end the diagnostic odyssey of this patient. So the challenge here is being a mosaic and being at low frequency or relatively low frequency. Next slide. This is an example of a missed variant in the mitochondrial DNA. As you know, the mitochondria have genes that are encoded with the nuclear genome, but also some with the mitochondrial DNA. And in this case, what we had is a variant in the mitochondrial DNA, which had not initially been looked at when doing the analysis. And we were able to find a pathogenic variant in gene MT-TL1 that also was at a lower frequency than the typical 50% that you were expecting for heterozygous variants, and this is due to the heteroplasmy that we have in the mitochondria. As you know, we have mitochondria with different genomes in each of the cells. Next slide. And the last example is an example of a missed CMV. Originally, this is a relatively small CMV of about 200 base pairs in heterozygosis, and now we were able to see it's on the gen APC, and this is from a family that, as you can see at the lower end of this pedigree, there is the haplotype that matches with the phenotypes of multiple adenomas in many of the individuals of this family, and therefore the diagnosis there was familial adenomatous polyposis, and thanks to the CMV analysis, dedicated CMV analysis in this case with clean CMV, we were able to identify this relatively small deletion that then again finalized the diagnostic odyssey of this patient. Next slide. So in conclusion, I think that this approach that we've been following in SOVRD highlights that it is very useful to do a systematic cutting edge research reanalysis, because the data pipelines are not harmonized across the different centers and across the different countries, and sometimes bringing in additional pipelines, looking at other variant types, or even trying different methods helps to discover need diagnosis. And also it is important to highlight that many of the diagnoses were not in the typical SNV and index that we tend to look at first. And also we wanted to highlight that this two-tier approach that we have developed with the bioinformatician expertise plus the clinical research expertise has been working very well in SOVRD, and I think it's a practical blueprint for reanalysis that can be used in future projects where high-scale reanalysis is needed. I think the next two slides, Paul, are just quickly to, if you can move to one, it's an acknowledgement of the co-author list of these first data sets I showed results from. And the next one is the full consortium authors from the ERNs. And finally, it's just a slide to end the presentation. So thanks a lot. And I'm not sure, Holm, if I'm taking questions now or at the end of the presentation.
Yeah, thanks, Sachi. So if you had any questions, please put them in the chat or just put them down somewhere or remember them once we come to QA, because we will be doing first the part of the meeting focused on the Solvaton concept. So also covering the second main approach we follow in SOVRD, and then there will be a Q&A session. Okay, so SOVRD concept. This is actually a novel concept, and it kind of puts into operation the two-level expert analysis and interpretation approach Sergio already described. So as you've seen, we have two main groups which collaborate, which are the data analysis task force, so data people on the one hand, and on the other hand the data interpretation task force, so the clinical and genetic people from the European reference networks, and they work together and come to respective disease-causing variants, the interpretation based on the analysis results from the data analysis task force working group. And the solvatons we did is mainly a process to really organize this kind of collaboration so that we can make sure that data people and clinical people and ex-users to the genetic people, which I think are really on both ends of the stick or both sides. This is the solvaton is really a process to organize this kind of collaboration. And it starts, of course, with the production and the release of the analysis results, which is being done by the working groups. And as Sachi has shown, we have working groups of different variant types, which did and are being doing the respective analysis. Then a on-site or online workshop is being organized, which brings both sides, so DITF and DETF together. It starts with a presentation of the analysis concepts and tools, as well as the results and the cohorts. Then there's an explanation of the interpretation workflow. We have the demonstration of exemplary interpretation of analysis results, and then there's hands-on work by smaller groups. And normally we did that organized in DITFs in disease groups, so to really make it work in a more focused manner. After this workshop, which resulted already in quite a number of solved patients' families, the work will be continued. So the continued interpretation work after the solvaton workshop, which is organized through respective communication means. So we have a communication channel, and we did that, and I'll be doing that via Slack or Team, but that is really not the point. So we need a communication channel, and then we have also defined consultation hours, and this entire process ends. So it doesn't go on indefinitely, but at a certain point in time, the interpretation work is being done, and then this solvaton focused on a respective variant type is at its end. So this is a picture which you will see once again, so I can go over it now. This is the agenda of one of the solvatons, so of the workshops we did. So you see the first day mainly dedicated to explanations and to demo, and then day 2 and day 3 are mainly focused to really do hands-on work on the patients and families, which we try to solve. And I think you understood that already. It's, of course, the main focus is on cracking cases, so solving unsolved cases and families. But there is, of course, also an educational aspect of this, and it goes into both directions. So it, of course, goes into the direction of clinicians, of clinical geneticists, which learn with respect to data analysis types, and also then how this is actually being depicted. And then, of course, it goes also the other way, which is the education of clinician towards data people, so the actual application of the methodologies for solving the unsolved. So we did 4 rounds of solvatons already. RNA-seq, then short read whole genome sequencing, long read whole genome sequencing, and optical genome mapping, and multi-omics. And we did it on-site, we did it fully virtual, we did it in hybrid, so it all works. But of course, it's best actually when people meet in person and have the workshop in person. And what you will now hear are 2 examples. So the first example will be on long-read sequencing and optical genome mapping, presented by Alex Heuschen, and he can say a bit more about himself as well. And then there will be a second example, and we get to that after the long-read and optical genome mapping. Alex, over to you.
Yes, thanks a lot, Holm, for the introduction. I'll indeed try to share my enthusiasm for how we approached these rare disease mysteries by the two long-read technologies that we applied, that is long-read sequencing and optical genome mapping. I'm a professor in Nijmegen, the Radboud University Medical Center, and I had the great pleasure to co-lead work package 2 with Sergi, whom you just heard. So if you go to the next slide, this is the excitement of the second day of our long-read Solvathon in Nijmegen. So this is the crowd that joined us on site, and there were additional 15 to 20 people that joined in the hybrid mode, so for some engagement online. And others continue to solve cases in communications via Slack and others, as Holm explained. So the two long-read technologies that we use specifically here in SolveRD are optical genome mapping on the left and long-read sequencing. And here we applied hi-fi sequencing by PacBio. And that is described for those that are not quite familiar on the right-hand side. I don't have time to go into all details, but my understanding is that you can access the slides later. So if you want to read up. In essence, the most important bit is optical genome mapping has nothing to do with sequencing. That really just is very high resolution cytogenetics, where you start detecting structural variants from 500 base pairs onwards, while long-read sequencing really gives you everything in terms of full sequence resolution of all variant types. And that is certainly emerging, and we were lucky to already do close to 500 genomes in SolveRD. And for both, I'll give you very brief summaries of what we've achieved so far, and in part thanks to the Solvathon. So if you go to the next slide, this is the ambition was to run 500 hi-fi genomes. At the time we conceived this project, and when we started data production, this still was overly expensive, so we only afforded modest coverage sequencing. So 10-fold hi-fi sequencing was what we get on average. And the data I can present today, we have sequenced a total of 480 samples, but we have a more in-depth analysis of the first 293 samples from 114 undiagnosed families. And if you go to the next slide, I just brought one example. We had two examples, both coming from patients with from the ERN NMD. Both had the suspicion of muscular dystrophy, but both had no molecular diagnosis, but now they do. And that is inversions that were identified by the long-read sequencing that break the DMD gene. Strikingly, in the lower case, the inversion not only breaks the DMD gene, but also a gene called NHS. That is also an OMIM registered Mendelian disease gene causing Nance-Horan syndrome. And in fact, the patient not only had muscular dystrophy, but also a cataract in the eye. And that is part what patients can exhibit with Nance-Horan syndrome. So in fact, this is a blended phenotype caused by disruption of two genes by inversions that these long-read technologies find now much more clear-cut than previous technologies. Go to the next slide. I brought a summary table. So what we identified just in this first batch of samples is in 13% of cases, we found a most likely genetic diagnosis. And we generated, I think, a quite conservative estimate of additional candidates that were largely also discussed in the Solvathon. All structure variants that are new have never been described, so the genotype-phenotype correlation is still a challenge, but we're following up upon those. And there, I think one major lesson is that the long-read sequencing in particular has strength to detect structure variants and STRs. Go to the next slide. Everything that I've shown here is already written up in a preprint, so if you're interested in much more details, I refer you to this medRxiv manuscript of our consortium. Then, next slide, switching gears here, going to the optical genome mapping. Should say that in part, some of the samples overlapped and reproduced not only optical genome mapping, but also the long-read sequencing or other omics. The ambition here is to have the data available soon for 300 cases, but this was the latest add-on to the SolveRD project in terms of its omics data generation. So what we have thoroughly analyzed so far are OGM data of 165 individuals from close to 100 families. You see the biggest contributor in here was Ithaca, and for some of those, we also included trio sampling with a much more straightforward de novo analysis, and that can be already seen on the next slide. So the benefit here is if we find a lot of rare events, potentially all of them could be interesting, but if a de novo hypothesis exists, then we can basically look at all the de novo proven de novo structure variants. And what we find here with the resolution that optical genome mapping offers, we find one de novo event in 4 genomes. So only 1 out of 4 trios presents with a de novo event. And here you see the 7 de novo structure variants that we identified in the 35 trios that were available to us as part of this endeavor. None of those are clear-cut diagnoses immediately, maybe unless the 7th case where we find some kind of chromotripsis. But I would say every de novo deserves follow-up, and by definition almost is a very good candidate for the respective disease case. If we go further one slide, for a lot of individuals, we didn't have the power of looking systematically at de novo events, but rather looked at all the rare events. That means with the resolution of this technology, we find around 50 per case. And then we overlap with the DITF expertise for the known and established genes. So basically there the question would be, can we identify a structure variant in a gene that was overlooked in the past and a well-established disease gene? But in some, of course, we can also discover novel events in yet undescribed genes. And here's a short list of, I would again say in a very conservative estimate, the best candidates that we identified. The 4 latter cases from Rita are also shown on the next slide, because those really provide a proper diagnosis already. If you click one more, there you see that we identified a 14.3 kb deletion in the SERPING1 gene that was a strongly suspected locus for both of these families. And in family 2, we identified an insertion. Both are cases presented from ERN Rita by Laura Battle-Masso from Barcelona. So those are 2 proper diagnoses achieved by OGM and discussed with Laura during the Solvathon. Next slide. Just to give you the very quick summary of what OGM delivers. I think this very nice and very clean data set allows us to very systematically look at de novo structure variants. By my strategy, all of them are very good candidates. And across everything de novo, but also inherited structure variants, we now have excellent candidates at 13.4% at this very stage. But as Holm explained, there is still some ongoing consultation hours. So potentially that can be further confirmed or maybe even grow. So I think that adds to the general note that structure variants may have been overlooked in the past and still play a very important role in so far undiagnosed rare diseases. Go to the next slide. Here I try to just summarize what we experienced based on our working groups, but also the Solvathons for both technologies, what the best clues are and the best benefits of both of these technologies. I think optical genome mapping has the benefit that it has a very low false positive rate. Hence, we can filter quite effectively to the true de novo structure variants. Also, we can very easily spot inversions and translocations. And in my perception, every inversion translocation, which are by definition quite exotic events, deserves thorough follow-up. And quite often that's an excellent candidate to dive in deeper. One other benefit of optical genome mapping is for people that have not so high affinity with bioinformatics, analysis is very intuitive, and the visualization of cytogenetic events is certainly favorable over any sequencing at this stage. And as I summarized on the previous slide, in 10 to 15% of cases, we generate extremely good candidates. So certainly a very good first shot of undiagnosed diseases. The long-read sequencing, there I would say the 2 biggest advantages and most successful variant types are structure variants and repeat expansions. And again, also here, whenever something is de novo, in particular a de novo structure variant or de novo repeat expansion, that directly prioritizes a variant to be followed up in greater detail. Then I'll close with one final slide where I just listed, because we anticipated some industry participation here in this workshop, some of the needs that we already identified. I think generally, especially the long read sequencing, but also the combination of optical genome mapping and any sequencing can still benefit from further improved visualization. So I think sequencing never comes with a cytogenetics mindset. So just looking at a VCF or an IGV plot is not sufficient to really make sense with a rare disease focus. I think there's still something to gain by doing assemblies, not just mapping, but assemblies of genomes by combining these 2 technologies that I presented. I think we clearly still lack databases of population structure variants or structure variants and SDRs in particular So their filtering for rare events is not as trivial as it is for point mutations, because all of you know that we benefit largely from NOMAD and similar consortia. So that has to be built for long reads, clearly. There is a need to also more genome-wide call for STR repeat expansions. That is not exactly trivial yet. Usually one uses a catalog, but tools are in development for that. And generally speaking, and I think Vicente will allude a little bit to that, we certainly generate overlap samples where we have produced multiple data sets, and the integration of that data is not exactly trivial, but very promising. So I think there's another need for us, but also industry partners to co-develop tools to make more sense of multi-omics approaches to rare disease. With that, I'll close by thanking everyone involved. That's stated here. It's our OGM working group and all the respective partners that share data for this presentation. And on the next slide, everyone involved in the long-read sequencing effort of SolveRD and its respective working group stated on the left column here. Thanks a lot for your attention, and happy to answer questions later in the Q&A. Thanks a lot.
Yeah, thanks, Alex. Very nice. So as I said, please remember your question or put them into the chat, and you will get the opportunity to ask your question once we have gone through the second example, which is on RNA sequencing. And Anna, Vicente, I hand over to you, and sorry for clicking already.
Yeah, thanks, Holm. Indeed, so as Holm and Sergio and Alex already say, within SolveRD we start with the DNA, but then we realized in many cases that that's not enough. We found many variants, especially whole genome sequencing. Some are hard to interpret, or some are we just don't find any variants. So we decided to move forward to this second layer and to perform RNA-seq in some selected unsolved cases. Next, please, Holm. And here the idea is that we collected, or it was collected at different hospitals, the ERNs, RNA-seq from different accessible tissues. Within SolveRD we have mostly blood samples, but also PBMCs, some fibroblasts, and for the NMD, for the neuromuscular patients, we have muscle samples, which was actually quite great because it allowed us to even test the gene expression in those tissues. Next, please. So we extract the RNA from there, and then what we detect, or our approach in rare disease, is to find this aberrant expression and splicing. This is different from differential expression, in which we are comparing two groups. But here what we see in the orange plot is the distribution of the gene expression of a specific gene. And there we see that for one patient in this population, which is the whole cohort of rare disease, for example, that the gene expression of that gene is very low for that sample. And that may be the disease causal one. It doesn't have to be. We have to associate it with a rare variant, and we have to prove that indeed the gene is, the phenotype of the gene associated to the one of the disease of the patient. But it can be indicative that there's something wrong in that gene that deserves further exploration. Then we also detect aberrant splicing, which is what we see on the right part of the second row. It's a way more complicated problem than aberrant expression. So it requires further analysis even from the software side, and the methodological side is also more complicated. And the idea is that we can detect all of these four cases, which are skipping of an exon that should be used, an exon being truncated, an exon being elongated, or even the creation of a new exon inside the intron that is not supposed to be. So we have algorithms that are able to detect both of these aberrant expression and splicing events that are different and are specialized from the ones that are algorithms that were developed to detect differential or alternative splicing. Next, please. And then there's many papers about this since 2017, more or less, that have shown that there's an increase, around 50% increase diagnostic rate over whole exome sequencing data. Next. And here what I would like to emphasize is the importance to first train the participants, because most of the geneticists from the different ERNs that we work with, they're used to, or they understand, right, what is a deletion, a missense variant, an allele frequency of 10 to the -4, or that it has not been seen, this splicing variant in GNOMAD. But they're not super familiar with what is the expression fold change or what is the delta upside of splicing. So that's why when we were designing the Solvaton, the RNA-seq, which was the first one we decided that it's very important to have first a training session of what exactly are we looking for here, how do we do it, and what do we expect to find, so that people more or less know how to interpret the tables that they were about to receive. Next, please. Now, so why do we do this RNA expression splicing? So expression allows us to, or in many cases, they are caused by this type of variants. First, variants in enhancers or promoters, variants in UTRs, short tandem repeats, of course, the usual loss of function variants like stop frameshift and splice site, and also very important structural variants, including deletions, duplications, translocations. So we have examples, not just from Solvardi, but from also other cohorts of each of these 5 types of variants that has led to aberrant expression in the disease causal gene. And this is quite interesting because it could have been that we already found the stop for frameshift variant or the UTR variant and we just want to validate it, or that we didn't find it at all, like a structural variant or a complicated inversion or even an STR, that the analysis was not performing the available data, and that through aberrant expression, we were able then to come back to the gene to analyze the DNA, and then, oh, indeed, we detect there was a duplication that we missed originally in the first analysis, and through this analysis, we found it back. Yeah, sorry, there seems to be something off there with the last exon. If you can go next, Holm. So also we detect aberrant splicing as well, and that has led to, for example, coding variants that cause exon truncation. It can even be a synonymous variant. We have examples of synonymous variants that activate a new splice site inside the exon, thus truncating it, and that is pathogenic. And usually we all agree that synonymous variants are not prioritized, so they might have been filtered out from the first initial analysis. Also, of course, the splice region variants, which is not the same as splice site in the direct dinucleotide, but like just in the region maybe 3, 5, even 7 base pairs inside the intron. And many of these that are still variants of no significance in most cases. They can cause exon elongation or exon skipping. We have examples of both, and that we can confirm with the RNA and then validate that indeed these variants in the splice region indeed cause missplicing. Next. And the last example, these intronic variants are quite interested. So in many cases, of course, intronic variants are also not prioritized. And if whole exome sequencing was performed, they were maybe not called. But we have found these cases of intronic variants that activate cryptic exon or create a new exon completely within the intron, and they contain a premature termination codon. And those we tend to see both with aberrant splicing events and with aberrant expression events as well, because of the premature termination codon that is contained there. And this usually, as I said, not called by whole exome sequencing, by whole genome, yes, but by whole genome not prioritized at all the intronic variant here. Next. And yeah, so this slide, something that I want to emphasize here a lot is RNA-seq results are only interpretable with DNA results. Otherwise, we just have some aberrant expression, but we have no idea what's going on. So only we say this aberrant expression comes with this stop variant, this aberrant expression comes with this application in that gene, it gets interesting. The same with splicing, right? This aberrant splicing event comes with the splice region variant. So it's very important that to combine both the RNA-seq and the DNA results, whether they're from short read, whole exome, whole genome, long read, and even optical genome mapping. We have had great results combining our RNA with all of those technologies. Next. So I'll just give one example due to time. This happened live on the first RNA-seq Solvathon. So we found that this gene, this NOP56 gene, was 50% expression reduction in 2 unrelated individuals. This, I mean, for me, a bioinformatician, this tells me not much, just like this happening, and this usually suggests a single allelic loss of function variant. But the corresponding clinician was there, and for her, immediately NOP56 rang a bell, because that gene is associated with this spinocerebellar ataxia that her lab investigates. So that rang a bell. This cannot be coincidence. So then also the person responsible for the analysis was also on site. He was able to run this expansion hunter, and we found these STRs, these short tandem repeats, in these 2 patients, and even in the father of the other patient, thus solving the case in less than 2 hours after presenting the results. And this, next please. Next home. So this advocates for the importance of the, oh no, one before, sorry. Just one before. Yeah, so this advocates for the importance of having everybody there in the Solvaton, right? The bioinformaticians, the clinicians, the people that do the DNA, the RNA, all possible experts, because this case was solved thanks to the 3 different people being there, and but they contribute their expertise with their findings, either there or running stuff, and we get to these very fast solved cases. Next. So overall, this was from the first RNA-seq hackathon. Everybody rated it as useful. Some people also think the RNA-seq results can greatly improve or help diagnosing the patients. And we got quite positive feedback. I think everybody was happy some way or another. Like, this is just not passive learning, right? The idea is not just to train, but like everybody's there to work, and you see that everybody was active. There was almost, not like Alex likes to say, no time to rest, even in the coffee breaks, right? They're half an hour, but usually they're only lasting 20 minutes. Everybody wants to go back because they found something interesting and to discuss, because you have the clinician next to, you have the DNA expert next to, the RNA expert next to. So in general, the environments are quite nice, and you get to meet the people that you have only previously heard by email or so. Next. So with that, I'd like to thank all the RNA-seq working team, and of course all the DNA as well, the different ATFs, the ERNs that contributed with data. I think we did quite a cool, like, this was the first one, the very first one in Barcelona, that was mostly on site. But also, as you can see, that we had online participants. It was the kickoff of all. I think due to the success, we have a total of 5 solved samples. There are many candidates. We were inspired to keep on doing more. Up to now, we have 20 solved samples. So 1 year later, we have way more, 20 solved samples from around 300 unique affected individuals with a rare disease. So we are in a 6-7% increased diagnostic rate, and it all started with this Solvaton more than a year ago.
2 slides. So, as said, luckily, this is not the end. Even so, Solvar-D finished end of March this year. So we're going to continue, and we're going to continue what we have been doing in Solvar-D now in EDERA. And EDERA is the European Rare Disease Research Alliance. And if you look at EDERA, it's much more than Solvar-D has been. So it's really the European rare disease research ecosystem for the years to come. And you see the numbers. This is really a huge initiative in which a large number of people will be participating. And even in terms of people initially integrated, it will be more than 1,600 persons. So it's really huge already, just the beneficiaries, and it's going to grow, I think, significantly. And if you look at the house of EDERA, so that's the internal structure. Then you see that there are a number of parts which are part of EDERA. So if you are familiar with the European Joint Programming for Rare Diseases, it's somewhat different, somewhat similar, but also only to a certain extent. So similarly to the European Joint Programming for Rare Diseases, it's got a funding pillar. So the joint transitional calls for collaborative projects will be also part of EDERA. And there will be at least one call on diagnostic research, so linking to what we will be doing in EDERA. Then there is what's being called a support structure, and there's, for example, a data service hub which we will be collaborating with. And if I say we, I mainly mean the Clinical Research Network, which is the in-house research part of EDERA, and within this in-house research part, there is a larger part. So 3 out of 7 work packages focused on diagnostic research. And if you look at the title of the work package, so diagnostic data availability, genome reanalysis, research pipeline, and innovation to shorten time to rare disease diagnosis, meaning long read and multi-omics, then you see that it's really a kind of a scaled up continuation of what we did in SOLVA-D. But here, to be complete, there are of course also within the clinical research network work packages on real-world data and outcome research, but also on therapy development, so ATMPs and NFU approaches. And what we do in the diagnostic research work stream can then directly be transferred to the respective outcome or also therapy development programs of Adera. And then the one which I didn't mention yet in terms of main pillars of Adera, there's one on coordination strategy and one on international capacity alignment, meaning that the focus of Adera is not only on doing research, but also on building infrastructure which can be used to connect with what is being happening at national level or even at international level, so that this is becoming really a international effort and an international program, even more so than already in SOLVA-D. Okay, this is just to give you a brief overview of the main objective which we have in the diagnostic research. So the clinical research network of Adera and then the diagnostic research work stream. So 3 work packages as said, one on data availability, including standardization and also the return of research results, then one on really on reanalysis, and the third one on complete genome analysis and on multi-omics approaches. And we have 3 main drivers. One is technological advancements, so from exome to genome to long reads in terms of also reanalysis. The second one is federation. So we have been dealing in SOLVA-D, but even more so we'll be doing with large data amounts in Adera. So we'll be moving from a more centralized distributed approach we used in SOLVA-D to a federated one. And the third driver is really an upscale in terms of what disease groups, which youth and FS networks are being involved. So we had 6 in SOLVA-D, and the aim is to involve all of them if there's a genetic diagnostic interest in Adera. Okay, and that's where we had to, and we will be starting in September and are now preparing the start of Adera and also continuing what we're doing in SOLVA-D. So I believe it will be a seamless transmission of what we do in SOLVA-D into Adera, but as said and as shown, with a significant upscale. Okay, I stop my slides, and again, allow questions from the participants and invite you, if you're interested, to get back to us. And that includes both, so interested in learning more about SOLVA-D, but also being engaged in Adera, because as said, Adera will be even more community-based, so that we would be happy to welcome further people. Then with that, I do thank my co-speakers at this webinar. Thanks a lot. I think we nicely showed what we've done and how successful we were in SOLVA-D. And I think that's a very good basis now to first solving more patients, but also for what we plan to do in Adera. Also in terms of, well, I think collaborative rule, this is a good basis. So as said, please get back to us if you want to learn more, if you want to collaborate. We're really happy to discuss and to talk. And with that, thanks again. Thanks for participation, and have a nice rest of the day. Thank you.